So I'm Ms. Burleson, and I'm going to introduce organic molecules to you. But my first question is, why can we not just live off of all the things I see y'all bringing in the classroom for snacks or for lunch?
We can, can't we?
No. Does anybody have an idea why that's a problem?
There's no vegetables up there.
I don't see any meat up there.
Okay, very good point. All right, we're missing a lot of very important nutrients. And it's not just that those nutrients are healthy and good for you, it's that they're actually the building blocks of you, of what makes you. So first of all, we need to talk about the 4 elements that make up 96% of your mass. 96% of you is these 4 elements. So carbon, hydrogen, oxygen, nitrogen. Now a nifty way to remember that is the word CHON. So obviously C is carbon, H is hydrogen, O is oxygen, and N is nitrogen. So easy way to remember, those 4 elements are going to be very important, so keep them in mind. Another term that's going to be important throughout this section is the word organic. Now a lot of you may think of the word organic and you think of, oh, it's organic, it's healthy, it's in the grocery store, they farmed it without chemicals. That is a use of the word organic, but for our purposes, organic means made of carbon. So anything organic, made of carbon. So that's why we call these the organic molecules, because they're made of carbon. All right, the structure of organic molecules is kind of like a puzzle. So think about putting puzzle pieces together. So they have monomers. Mono means
One.
Mono means one. So one piece, one subunit, put together making a polymer. Poly means
Many.
good. Poly means many. So when we put many monomers together, we make a polymer. So again, think of it like a puzzle. You've got one piece of it, that's the monomer. You put it together making your puzzle or making your organic molecule, that's your polymer.
All right, hi, I'm Ms. Hall, and I'm going to continue what Ms. Burleson was just telling you guys about. I'm going to tell you the first organic molecule that we are going to talk about, and this is carbohydrates. All right, I know you guys are familiar with the word carbs, but we're going to break it down a little bit for you. Those 4 elements, carbon, hydrogen, oxygen, and nitrogen. Out of those, we're only going to be seeing 3 in carbs. Carbon, hydrogen, and oxygen, all right? And like Ms. Burleson was saying, you have monomers and polymers. With carbs, we like to call them something special, and we call them saccharides. If we have just one saccharide, we call it a monosaccharide, and this is the building block, like a brick. If we have multiple, we have polysaccharides. Remember, poly means many. We also have something called disaccharides. Di means 2. Good. So we have monosaccharides, which is just one saccharide. Disaccharides, 2. And then poly is many. And this fancy word saccharide, it sounds weird, but it just means sugars. All right, so when you think of carbs, I want you to think about sugars and starches. Does anybody know some examples of carbs that you might think?
Pastas.
Pasta, yeah. Bread, good.
Sugar.
Yes, sugar. Good, all right? So this is what we're going to be looking at. There's some really, really awesome examples of monosaccharides, disaccharides, and polysaccharides. Our first one, we have glucose. That's really, really big when we're talking about photosynthesis in the future. Glucose is made by plants. That is a monosaccharide. It's one sugar. Disaccharides would be sucrose. And polysaccharides, we have cellulose. All right, that's really important for plant structure in our cells. Okay, so the way I like to remember it is if it rhymes with gross, because sugar's gross and you don't need to eat a lot of it, it's a sugar. Glucose, sucrose, cellulose. All right? Alrighty, so here's some examples. We have this lovely picture, just like you guys said, pastas, bread, we have corn starches. The function of carbs. Why do we need to eat carbs in the first place? Why is this important part of our diet? It's a source of quick, short-term energy. Now that's important that it's quick and short-term. And again, examples are bread, sugar. And the way I like to remember that it's short-term is that I coach swimming, and before a big meet, we will what we call carb load, right? We eat a ton of carbs, a ton of pasta and bread and stuff because we need a big source of energy that we're going to use up really quickly during the meet. All right, so I like to call this the Snickers bar. You know the commercials where it's like, you're not chewing, you're hungry, and it gives you a quick burst of energy, that Snickers. It's sugar. Let's go test it out. All right, so we're back and I'm gonna show you, I was talking about carbs earlier, remember that's your starches and your sugars. And I'm gonna show you how we would test certain foods to see if they have carbs in them. All right, Miss Wance is gonna be my lovely assistant and she is going to show you what to do and I'm gonna tell you what she is doing. But remember, the first thing you gotta do is put your goggles on.
Oh, darn. Okay.
All right, so we have 2 test tubes here. We're gonna put only one food in each so we know where we're testing just that food. Okay, so for the first test tube, it looks like Miss Wayne is gonna put a piece of an apple inside with her little forceps. She's gonna squeeze it down in there. She's also gonna put Benedict's in there. Benedict's has this really pretty aqua blue color. So she's just gonna cover up the apple and she can set that aside so we can make the other test tube with our other food. I wanna make sure that you guys know what a positive test looks like and a negative test. So the other food we're gonna put in here and test for a carb is egg. This is a boiled egg that's gonna try and go down the test tube. All right, a little more Benedict's on there. Now remember, we're looking for carbs and it's really special because carbs has 2 tests. One for sugars, which is Benedict's, the one we're doing now, and then another test, iodine, that test for starch. We'll do that in a second. Okay, the most important part is that you have to heat up Benedict's for it to work, okay? So we have our test tubes. Miss Wance is gonna use her test tube holder and she's gonna let it heat up just for a minute. All right, so now that our Benedict's has heated up for a couple of minutes, we can definitely see that there's a color change happening. That's our reaction, okay? So if it's positive test, meaning that there is carbs, specifically sugars, in this food item, it will change colors. And this is an example of a positive result. It kind of looks like tomato soup. It'll change like to a greenish color and then an orange red. Our negative example, right here, didn't change any color. It's still that aqua blue. Now, yes, it looks like it might be a little green or whatever. That's just a little bit from the food. That's not actually a positive result. It's still really blue. You would see a very big difference. All right, so we're back again. Still looking at carbs. Remember, starches and sugars. Remember, it's got 2 tests. So our Benedict's tested for sugars. Today we're going to do the iodine that tests for starch, okay? So Ms. Hunt is my lovely assistant. Make sure she's got her goggles on. She has a Petri dish here that she's going to put her food samples on, and we are going to take a bit of a Ritz cracker, potato chip, and some egg. Remember, again, this is boiled egg, so this is the yolk part. That's why it looks like that. An important part here that you see, she is placing the food items in the Petri dish on top of a white piece of paper. That is because iodine itself is really dark and it would be really, really hard to see the actual test results on top of our black lab desk. So we need the white piece of paper to be able to see really well. All right, so Ms. Hunt is gonna put 1 drop of iodine on each food item and 1 in the middle so that we can see what iodine looks like by itself. We wanna do this so we have a control to see what iodine looks like without reacting to anything, so we can see if it was a positive or a negative result, okay? So with our Ritz cracker, we know it's made of flour and all that stuff. It's bread-ish, okay? So this is starchy and it's got that positive result. You're gonna see a really, really dark black color, okay? If we look over our potato chip, we also see a really dark black color, especially if we're looking in comparison to the middle, okay? Again, potatoes, that would be your starch. Over here in the top left corner, we have our egg, and you can see that on top of the egg, it's still kind of that orangey color, especially compared to the control. It's not actually changing anything. There is no starch in the egg, so that would be a negative result. Oh, sorry.
Now that we're done, you gotta throw away all the food.
No, Miss Hunt, you don't want to put it down the sink. You need to put it in the trash can. Make sure you guys put this in the trash can. Don't clog up our sink. Gross.
Hi guys, I'm Miss Hunt and I'm going to teach you guys about lipids. Alright, so lipids, the elements that we're going to be looking for in lipids are carbon, hydrogen, and oxygen. The monomer, the single unit for a lipid, is a glycerol and 3 fatty acids. And here's our lovely picture of what that looks like over here. The function of your lipids, your fats, is long-term energy, insulation, and they're also a big component of your cell membrane. We're going to talk about that here coming up in biology. Examples: butter, oil, steroids. carbohydrates, cholesterol. All right, all these oily things, they're a lipid.
Let's go test it out.
Now, your last test is for the lipids. And this is actually a very simple test. You don't need any fancy chemicals. In fact, you've run this test anytime you've gone to McDonald's and you've come back with that greasy bag and you've noticed that it's translucent. So, here we have our paper. And if you were to actually run this test, you would actually take the food and you would rub it in each area that is labeled for that food. Now, we've already done it for you because it takes a while for the liquids to dry, and because of movie magic, I can show you that this is a positive test for lipids in chips. You can hold it up to the light, and even if you put it down on the dark lab bench, you can see that definitely chips are positive for lipids.
Looks like my cookout bag.
Yep.
Hey y'all, I'm Miss Weatherby, and I'm going to be talking to you about nucleic acids. Now, for nucleic acids, this is a really important part of biology, but we're going to talk a lot about it later on. But for now, we're just going to learn two parts of it. One, the structure, and the other is the function. Now, nucleic acids, they have 4 of the elements that we talked about before: carbon, hydrogen, oxygen, and nitrogen, but it also has phosphorus, okay? And phosphorus is in our phosphate group, which is part of our nucleotide. So our nucleotide is the monomer. So whenever you want to know the monomer for nucleic acids, you guys say nucleotide, okay? So our nucleotide is a little piece of a molecule right here. You have your phosphate group, your pentose sugar, and your nitrogenous base. And that hooks together and makes your nucleic acids, which is your polymer. So after that structure comes together, and it's actually kind of in a ladder here, and we see that's what you know of as DNA. Okay, and so DNA is the examples for our nucleic acid, and the purpose of our DNA in our bodies is for your information. This is where you get all your genetic information from and how your cells know what to do. Is that good?
Hi, I'm Mr. Caldwell. I'm going to be talking about proteins. So, like nucleic acids, proteins also have a unique element in nitrogen. They do not have phosphorus, though. So they have carbon, hydrogen, oxygen, and nitrogen. Now, the monomer, which is the simplest unit, or the most basic unit of protein structure, is the amino acid. Now, with nucleic acids, remember, DNA and RNA are responsible for storing and transferring that genetic info. So the ultimate goal is to make those amino acids, and the amino acids that are made will eventually be transported throughout the cell and then used to make proteins. So in everyday life, you hear about proteins being used. So athletes will take protein powders to get big and strong. What's actually in those protein powders they use are different types of amino acids, and those amino acids they use are what they call essential amino acids. And the essential amino acids are amino acids that you actually have to consume in your diet, meaning that your body cannot make those amino acids. Now, there's some amino acids that your body obviously does make, and that's what your DNA and RNA, they translate those amino acids and produce them for your body. So the monomer, to recap, is amino acids. Amino acids are joined together to make something what we call a polypeptide. Polypeptide is just simply many amino acids linked together. Remember, poly means many. So the functions, there's obviously lots of functions to proteins. Ms. Wance in the next slide will talk about enzymes. The function of enzymes is to speed up chemical reactions in the body, and they do that by lowering the activation energy. Proteins also provide all sorts of structure inside the body, whether it be in the cell membrane, and then the next unit we'll talk about lipids and proteins and how proteins are embedded in the cell membrane, and then we'll also talk about how proteins help in transporting stuff across the cell membrane. There's also some proteins called actin and myosin, which help actually make your muscles move. When you do like a curl of the bicep, that's your actin and myosin moving. And then there's proteins in all sorts of foods. So in that first slide when Ms. Burleson was talking about you have to think about your diet. Proteins are important, and they can be found not just in meat, like you commonly think, they can actually be found in vegetables as well.
Let's go test it out.
Okay, the next stain up is the Biuret stain. Biuret's going to be used to test for the presence of proteins. So how it actually works is that it interacts with copper ions and peptide bonds. So peptide bonds are what links to amino acids. So the Biuret's really going to recognize the peptide bond. So the darker the stain, the more peptides bond. So that correlates with more proteins. So the stain, as you can see, is like a light blue. When you test for proteins, you're actually going to look for a violet color. Otherwise, it's going to be not positive. So, Miss Weatherby is going to be doing the staining. First, we have a chunk of apple.
I can't do this with this.
A cracker. We're going to put a drop of milk down. And Miss Weatherby's going to add a drop or two of the stain to each of our specimens. And then she's also going to put a control drop down in the center. So again, remember we're looking for the presence of the violet. And you can see with our milk, right around the edge, you can see the violet color compared to our control where you still have it blue. So that would indicate a positive test for protein.
Hi, I'm Miss Wance, and I'm following Mr. Caldwell. And as Mr. Caldwell talked, he talked about proteins. I'm going to talk about one particular type of protein called enzymes. So enzymes are a type of protein. All of our enzymes will end in ASE. And really important, enzymes are very specific as to what they act on. For example, we talked earlier about sucrose is a sugar. The enzyme sucrase breaks down sucrose. Sucrase doesn't work on anything other than sucrose. So they're very specific as to what they'll act on. We talk about it as a lock and a key. Your key, does it open your friend's lock of their house?
I hope not.
No. So your key is specific to the lock of your house. What the enzyme acts on. So we have sucrose. Sucrose is the sugar. Sucrase breaks it down. Sucrose would then be the substrate. It's what the enzyme's acting on. Where the two join together is called our active site. Now, the main function of our enzymes, guys, is to speed up chemical reactions. How do they do that? They do that by lowering the activation energy. You say, what is activation energy? Activation energy is the energy needed to start the reaction. So I have a little graph over here. This is our energy. Without the enzyme, look how much energy is needed to start that reaction. With the enzyme, notice we don't need as much energy. That reaction is going to start sooner. So it causes the reaction to occur quicker. Very important, two basic keys, two characteristics of enzymes that you guys have to know. Enzymes are reusable. They can cause a reaction with one substrate, be used to do it with the same substrate, the same type. They are not used up in the reaction. They, however, can be affected by things. There are things that can destroy them. pH levels. If our pH levels become too high, they become too low, the enzyme doesn't work. Those enzymes have a pH that is optimum for their use. Heat will denature it. It will change the shape of the enzyme so it no longer fits into the substrate. And also inhibitors, they can block that active site So the substrate can't join to the enzyme. So those are enzymes. Enzymes, very important, they're found in the body.